So for the last 10 weeks, I've been here at TU Delft in the EBT department, working with Heiss, Berber, Damie, and David, all focusing on the application of phototrophic non-sulfur purple bacteria for wastewater treatment. And so before I start, I just want to go through an overview of what traditional wastewater treatment is. So traditional wastewater treatment, of course, we start with the wastewater that is then transferred to the water treatment plant. And for this demonstration, we're just going to focus on the organic compounds present in the wastewater. And then, so traditionally, aerobic bacteria is used. And for aerobic bacteria, we need aeration, which requires a significant amount of energy. And then when the bacteria metabolize the organic compounds, they are consumed, and this then produces carbon dioxide, which is a waste in this process. And then so the main product of this wastewater treatment, of course, is decontaminated water, but we also have a large amount of sludge that is produced, which is concentrated waste material. And so ideally, we would like to have lower amounts of sludge. And then this sludge is incinerated, which then produces large amounts of carbon dioxide. However, sludge is often recycled for application in agriculture. Then both the consumption of agriculture products and this decontaminated water is used by humans, and then humans produce more wastewater. So it's just one large cycle. And so with this process, we address many problems. The first problem that should be addressed is wasted organic compounds. The bacteria consume these organic compounds and produce a lot of carbon dioxide. But these organic compounds could possibly be used to make other products and be recycled for other processes. So we would like to actually utilize these organic compounds. The second problem is that there is a lot of carbon dioxide produced in this process, and we would like to minimize that production as well. The next thing is that there is also a significant energy requirement because of the aeration that is required for aerobic bacteria. And lastly, there is a large amount of sludge production. This is a large amount of waste, and it's unnecessary to have this much waste. And so in order to solve this problem, we can utilize anaerobic wastewater treatment. Anaerobic wastewater treatment, in comparison to aerobic wastewater treatment, does not require aeration because it doesn't require oxygen. There's also a significantly lower amount of sludge production, which is great because that, again, is a waste product. And then lastly, there is significantly less cost associated with this because there is very little energy cost because there's no aeration needed. And so how are we going to do this? We're going to do this through anaerobic digestion. Anaerobic digestion involves many steps, starting with these complex polymers and ending with methane, and then that methane is combusted to carbon dioxide. And so what we have in the middle of the process are these intermediary products, these organic compounds that contain but are not limited to propionate, butyrate, malate, and things like that. And then these are also converted to acetate. So if we want to minimize the production of methane and carbon dioxide, then we should utilize these intermediary products. And we can do that with the application of bacteria. And so ideally, this bacteria is implemented for anaerobic digestion. The bacteria can consume the intermediates and acetate. However, this bacteria will also need to use some of the energy obtained from these intermediates for its own energy production to sustain itself. And then with the rest of its energy, it can produce a biopolymer or other products. However, we want to optimize this process. And overall, this process will decrease the production of methane and carbon dioxide. But we want to optimize this process. We can do that through implementing phototrophic bacteria. So the process starts the same way. The phototrophic bacteria can consume these intermediary products, but it obtains a lot of its energy from light. So it won't need to utilize the energy that it obtains from the intermediates for its own energy. And then it will still produce the biopolymer, but the production of the biopolymer will be significantly increased. and this at the same time will decrease the production of methane and carbon dioxide. And so this is just an image to summarize what I just went through. This is energy use with bacteria that are non-phototrophs. And so we can see that they give off some of their energy to sustain itself and then produce less product than a phototroph would, because a phototroph would get its energy from light. And so now we need to determine a bacteria that would be applicable for this anaerobic digestion and optimizing the recycling of these organic compounds. And so we found that in phototrophic non-sulfur purple bacteria. This bacteria is commonly referred to as very metabolically diverse because it can metabolize and consume a wide array of organic compounds. And it's also a facultative anaerobe, meaning it can operate either anaerobically or aerobically, making it even more diverse in the way that it can sustain itself. And lastly, it is capable of producing PHB, polyglucose, and possibly other products that could be recycled and used for other applications. So it's a really great candidate for bacteria that can be used for this process. And so that takes us to our summer projects. This summer I had 3 main goals. First was to enrich a sample of PNSPB. The second was to maintain the SBR reactor and to collect data from the reactor. And the last one, the overarching goal, was to obtain a pure culture of the dominant organism in the sequencing batch reactor. And so to start off with, the first goal was to obtain an enrichment of PNSPB. So we use a sequencing batch reactor. As you can see here, this is the reactor towards, it's a very recent picture, very rich in bacteria. So the way the SPR works is that the bacteria will receive growth media in sequences. Specifically for our application, it was every 8 hours. The media it was based off of was an acetate media, and it received infrared light as its light source. And so going further into the growth conditions, it was growing in anaerobic conditions with argon in the headspace, so that we, again, tried to create that anaerobic condition. Our carbon source was acetate, and this was a very selective factor because not a lot of bacteria can utilize acetate for their metabolism, which would, again, select for PNSPB. Thirdly, our light source was infrared light. Again, this was another selective factor because other bacteria, specifically oxygenic bacteria, can utilize other sources of light, and we wanted to limit that as much as possible. So again, we could select for the bacteria that we wanted. Lastly, our nitrogen source was ammonia. We used a phosphate buffer, and we had systems in place to maintain a pH of 7. So this is a schematic of the reactor. You can see that we monitor for pH, temperature, dissolved oxygen, and redox. Everything is computerized, so our schedule of when the reactor receives its media is all computerized based on a schedule. In the sequential batches from our stock, we have argon gas in place to ensure that the headspace is always full of argon, and we do have acid and base pumping into the reactor to ensure, again, that we do have a pH of 7. And this is a diagram of the schedule that we have. The schedule runs 3 times a day in an 8-hour cycle for 24 hours. So we begin with the discharge ideal and feeding for 15 minutes when the bacteria receives its acetate, its growth media. And then for the next little over 4 hours is the reaction time when the mixer is running, the bacteria is feeding off of those organic compounds. And then for the next 3 hours is the settling period when there's no mixing, the bacteria are allowed to settle to the bottom of the reactor. And that brings us to our second goal, maintaining the reactor and monitoring it for its results. And so here we can see the progress of the growth in the reactor. We can see at the very beginning, May 29th, the bacteria, or at least our color of our bright red purple bacteria, was really not growing to the extent we wanted it to, because it's just the beginning. But over time and all the way to August 14th, just last week, we do have this dark red-purple growth, which is a great sign for us that we have selected for the appropriate bacteria. We'd also see here, especially on July 11th, I'm not sure how well you can see it, but we do have significant growth on the walls of the reactor, and that further indicates to us that this is definitely a phototroph, or likely a phototroph, because it is thriving off of that light. While I was here, I also investigated a lot with microscopy, and what you can see here is that we have selected for a clumping bacteria that likes to settle together, but what you cannot see is that the bacteria are very motile. They do swim around a lot in that sample. And then I also wanted to highlight the spectrum. Every day we would run a spectrum of the sample in the enrichment culture, and with every sample we noticed we had 3 peaks. One at 460, 800, and 860 nanometers. And what you can see here is that the blue line, this represents the spectrum at the beginning of the cycle, and the red line represents the spectrum at the end of the cycle. And you can definitely see an increase in the absorbance. This does not necessarily indicate an increase in biomass, but it does indicate an increase in pigment. And just so we can see a comparison from literature values, this is a graph that was taken from a piece of literature that did look at another species of Rhodospermonis, and we do notice very similar peaks. Our peak is around 860, their peak is around 870, we had one at about 800, they have one at 805, and so on. You can see that we have very similar peaks, which is very encouraging for us that we do have a bacteria of the species, possibly. And so overall, the overall project goal of using this bacteria is to enrich for this PNSPB. My specific goal was to procure a pure culture of the dominant organism in the reactor. We don't know for sure if it is PNSPB, but we wanted to, like I said, produce that pure culture of the dominant organism. Which brings us to our final goal, obtaining a pure culture. And so when we were obtaining this pure culture, we set up a batch environment, as you can see here. We would have our light source separated by a red film that's selected for infrared light that would then hit our bacteria samples. We had 2 different environments, one which was outside in the lab, you can see so here, but we also had another one that took place inside that we could, control for temperature. So the first experiment that we set up was an agar dilution series. The goal of this was to obtain single colonies. The medium of this was 2% agar and based on acetate, so exactly what is in the reactor. And as you can see here, we were very successful in obtaining those single colonies, which was the main, like I said, the main goal of this experiment. And so these 2 pictures were taken a couple days apart, and there are a couple things that I want to talk about. First off, you'll notice that the 8th dilution is missing in the second picture, and that's because it was used to streak out agar plates, because we did have encouraging single colonies in that tube. The second thing I'm talking about was more of the phototrophic activity that was going on. You can see here that the bacteria are thriving when they're exposed to light. This came directly from the tray, and you can see that T-shaped outline. And the last thing I want to say, overarching, that was successful about this experiment is that we did achieve single colonies. So the second experiment that we did was a liquid dilution series. These were inoculated with samples directly from the reactor. You can see that the 0 dilution is actually 10 to the minus 2 because we started at a higher dilution than normally. And so there's a couple things I want to talk about with this one as well. First off, 4 and 5 are not pure cultures. We can see that because 3 and 6 demonstrate the similar reddish purplish color, and these 2 both exhibit a reddish, a yellowish orange color. And the second thing is that initially dilution 7 did not include much growth, but after a couple of days you can see up here it's more zoomed in, and that we do have more significant growth, mass collecting at the bottom of our purple bacteria. And so the next experiment we did was just a general experiment to see how the bacteria would react to being exposed to various organic compounds. And so you can see here they were exposed to lactate, pyruvate, fumarate, succinate, ethanol, malate, butyrate, and acetate. And you see that most of them were very successful in growing in these media. However, you will notice that pyruvate does have a significantly lower pH, and this is just an experimental error because we were not able to ensure that the pH originally was 7. And so the next experiment we wanted to do was to find an appropriate buffer. We wanted to ensure that our batch systems were being buffered appropriately at 7. So we compared a phosphate buffer and a HEPAS buffer at different concentrations. And you can see here that the HEPAS buffer at 50 millimoles was very successful, not only maintaining the pH at 7, but also creating a really opportunistic environment for the bacteria to grow. And the next experiment we did was kind of a combination of the last couple of experiments, just as an overarching test before we started going forward with more pure culture attempts. So we combined the HEPAS buffers and anaerobic and aerobic conditions, and we inoculated it with the liquid dilution number 6. And as we can see here, the number 5 sample with the standard acetate medium with the HEPAS and malate in addition really provided a great environment for our bacteria to grow. And this experiment really helped us moving forward, identifying malate as a really beneficial additive for this bacteria. And so now is the time that we really went forward to streak out several agar dishes to attempt to get that pure culture. And what this graph shows is that we did a variety of environments. We had 3 different plate mediums. We had acetate, malate, and lactate. We also had various inoculum from a single agar colony from agar dilution 8. We used the liquid dilution number 6. And we actually used a sample right from the enrichment, right from the reactor. And we also varied the conditions from aerobic and microaerophilic. And so after that, we had many plates, as you could see from that experiment. And so we identified our most successful plates, our plates that showed most encouraging results of pure cultures, and we streaked those further. You can see these are all for secondary streaks, and we renamed them R1 through R5. So for R1 and R5, we started with a single colony from the 8th agar dilution, and they were streaked on agar plates with different medium. And then for R2 and R3, these were actually taken from 2 of the agar dilutions. They were taken right from the top of the dilutions because we noticed that there was a lot of bacteria accumulating right on top of the agar, probably because they were exposed to a little bit of oxygen, which helped that growth. After all, because they are the facultative anaerobe. And then number 4, we have a liquid dilution number 6 that was streaked onto a malate plate. And so we do have 5 different streaks right now, and so I'm just going to go into the history and details of 2 of them. So the first one is R1. Again, this was first taken from the reactor. Then we produced an agar dilution series. We took it from number 8 to receive a single colony, which was then streaked onto a malate plate kept in aerobic conditions. And then it was streaked onto this R1 plate, which is based on a malate medium. You can see the results here. We do have very successful growth of this reddish pigment. And then for R4 again was taken right from the reactor. Then it was streaked from dilution number 6 onto a malate plate kept in aerobic conditions, and then streaked one more time onto R4, which is a malate plate. And you see here, we do have many single colonies on this plate, which is really encouraging. And then so for our next experiment, we wanted to look at how our 1 through our 5 would behave in liquid media. And the media was based off acetate, malate, again, because we know that that is something that this bacteria would really thrive on, and the HEPAS buffer. And so the first thing I want to address with these liquid dilutions is the first one is that R4. You notice right away we did 2 liquid samples for this medium and taken from 2 different locations on the R4 plate, and we got 2 very different colors. What we can conclude from this is that there is a contaminant in R4, so it is not a pure culture at this time. And then the second thing I wanted to address was the growth rate. Initially, we saw lots of growth in R1, R4, and R5, but little to no growth, no noticeable growth in R2 and R3. However, after just 2 days, R3 saw significant growth from what looks like nothing to a very considerable growth. And this was generated from agar dilution number 9. And however, we also have R2 in the same amount of time, but did not grow noticeably at all. This came from agar dilution number 10. And so after we had done all of these streakings, we wanted to once again revisit our experiment with testing various organic compounds. You see here we tested similar compounds to which were tested before. And these were inoculated with the R41, so again, it's important to mention that this was the contaminated liquid media. It was kept in excess phosphate excess phosphate conditions with the HEPAS buffer, and all of them contained 5 millimolar concentrations of that organic substrate. You can see that all of them were very successful in growing the bacteria, with the exception of ethanol, which just means that this bacteria does not grow well with the ethanol. And then so one of our last experiments was exposing samples of the bacteria to a light versus dark scenario, where we exposed it to 8 hours of dark and 16 hours of light based on a timer. And after a couple days, we noticed that there was very little pigment shown. This is likely due to the fact that the bacteria are conserving their energy, so instead of making pigment, they're sustaining themselves. And so for the very last experiment, we did a final streaking. We did a streaking on malate and acetate plates, and at this time, we had 7 possible pure cultures. And so now I can share our results. We can see here that we have 7 possible pure cultures, very likely pure cultures. All of them demonstrate that reddish, purplish growth on these acetate malate plates. Again, we used malate in addition to acetate because these bacteria really do enjoy that. And so yeah, and here we are. We do see that we have very many single colonies, which is very encouraging for future holdings of these bacteria and to preserve them. And so overall, after doing all these experiments, after successfully gaining 7 possible pure cultures, we can look back into our implementation of this bacteria. We are not 100% certain that we do have this PNSPB until we get the DNA results back. But should this be that bacteria, we will successfully be able to apply it for anaerobic digestion so that it can obtain these intermediates in acetate, photosynthesize with light, and produce increased amounts of biopolymer and decrease waste, which will overall make this anaerobic wastewater treatment more sustainable and environmentally friendly. And that's my presentation. I'd like to thank all of you for your help and guidance this summer. And yes, thank you very much.